So the next challenging question is how to detect the virus. So as we said, the simple one is crystal violet to see the cytopathic effect. And here also I'm showing some images. You can see clearly after virus infection, you can see with these different staining methods. And also you can go a little further and you can also stain the infected nuclei also by using the different antibodies. And also one of the method is ELISA method, like you take the suspension and you can also measure how many virus particles are there by using the antibody coated plates. Basically it's a quantification. Okay. The other one is very simple. We all read this in our masters and graduation, like hemagglutination assay. When you add RBC, there is no virus, there is no reaction. And when you add RBC to the virus, it will agglutinate and you form a hemagglutination spot. But if you add antibodies against this virus, so that it will not allow to attack the RBC, so that there is no hemoagglutination. So it's very simple way of again measuring the detection of the viruses. So if you are more interested, there are different types of cytopathic effects, and depending on the virus also. Each virus has its own way of growing and also affecting the cells. For example, let's take this paramyxovirus. Basically, it is forming the syncytium. If you see the image, each cell has a more than one nuclei. Basically, this is called as a syncytium. Syncytium means a fusion of different nucleotide cells in form a multinucleated one cell. Basically, it is fusing the different cells. That's why you can see clearly it's a multinucleated syncytium. The other one is poxvirus. It has a cytoplasmic inclusion bodies. You can see the inclusion bodies where arrows you can point out. Then the other one, herpes virus, it has a nuclear inclusion bodies. The other one, adenovirus, has a grape-like structures. So that means the take-home message is each virus has its own way of damaging the cells. Some cells will damage the nuclei and also membrane and also material, genetic material. So it is very specific to type of virus you are growing. Okay. The next topic I'm moving to is detection methods, like viral detection methods. So the more relevant is COVID-19, but I will try to do the brief on each type of technique. So let's take, for example, COVID as an example. We are developing, people are developing different ways. Like, see, the test is depending on the type of analyte you are using. For example, if you are going to analyze the genomic material, RNA, so you go for the RT-PCR or CRISPR, also NGS, like genomic method. But the analyte is different. For example, you are looking for the surface receptor protein antigen, for example, spike protein. Then you have to go for the ELISA based method, or for example, we call rapid method, antigen based method. So the type of analyte you are measuring is very important, and based on the test will be planned. Okay, here we are measuring two analytes very importantly. One is genetic material, that is RNA from the virus. The other one is surface receptor antigens. And for example, if you have a virus in the body, it will induce the humoral response, like your body will respond and it will produce the antibodies. The antibody production will take place only after 7 to 8 days of infection, right? But here we are using two types of antibodies, that is IgM and IgG. IgM is a early antibody, and that shows that still infection is on, but body is reacting. But if you measure high amount of the IgG, means I think IgG is a kind of recovered patients. You see more amount, like a body is already completely killing this virus by producing the neutralizing antibodies against the spike and any other surface antigen. So basically, IgM is early antibody that indicates that infection is still on, but IgG will tell you that infection is completely gone. And that's what people are developing, the rapid antigen and antibody kits in the market. It's very simple. It's a lateral flow assay. You take some strip of paper or any nitrocellulose membrane. You put your antigen or antibody, whatever you are going to measure, and then you have controls and also synthetic proteins and RNA also you can use. So you have a control, positive control, and also you have your analyte, and then you add the serum. Serum is a very good option. And then if there is a antibody, it will cross react and it will give the color. It's a very simple one. And then coming to the very gold standard method is RT-PCR. I'll show the video for that also. So you have a single strand RNA in the virus, and we are converting to cDNA, and then we are going for RT-PCR method, because here we design the primers, and also we have probes, Tagman probes, let's say. So they're very specific. So they bind to specific regions. For example, in COVID, we are using E gene, envelope gene, as a screening gene, and ORF1A or N gene or RDRP gene are the what do you call confirmation genes. So there are different companies are just using the same primers and probes, and they're adding different fluorochromes. Some are adding what do you call that, FAM, GEN, WIC, and different types of fluorochrome they're using. For example, NIV kit has 3 genes, but there is one more kit called LabGun. It's from Siemens company. It's very good, and it also has but each company is using their different genes, but ultimately they are measuring the screening gene and confirmation gene. And the next one is what do you call that, nanopore sequencing. If they want to know the complete sequence, because this is very small, like it's a 30 kb virus, it's very small for NGS. So there are people are also using the NGS method to detect the virus. Different strains are available. For example, let's take in our population, in our country, we are even seeing this reinfection, second infection. But I think that indicates that the strains are also different. Some people are in low risk, some people are in high risk, depending on the virus strain and also host immune response. I think we all respond differently. That is where the whole mess is happening. Some people are asymptomatic, some people are mild and very severe. And severe, we mean severe also we have a ventilators and non-ventilators, and some are having sepsis. So this is very heterogeneous and infecting, showing the host immune response very differently. But this is not dealing with any method, with detection method. I'm just explaining a little more here. So detection methods are only based on the RNA and the protein, like spike protein and antigens. I think the CRISPR is too big to go because nobody using, but we can still do this. We can modify a particular region and we can design we can amplify, we can use the primers, we can detect this, but nobody is using this CRISPR-Cas9 method now. Yeah, I can show you a simple RT-PCR test, how we are doing in the lab. This is a final video so that we can imagine. I hope I have a video. Yes.
COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx, then it is rotated.
I think this is very important. I think most of the false negatives are happening with this problems in swab. So I think you have to reach this level where your nostril, what you call nostril, right?
For a specified period.
So if people are taking from a very how do you call that
time layer, to collect secretions that contain the virus.
So you are not going to take the enough tissue and enough viruses so that we'll tell that the patient is negative, but in fact the patient is positive. So this step is very, very crucial. I think what I'm proposing is sometimes we take both. You take the nasal and you can take the oral, and you can mix in the viral transmission medium so that at least you can get in one of the method. Otherwise, this is the biggest problem in current scenario, that people are saying that I'm negative, but after some time they're developing the symptoms and also they're collapsing. So
once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction (PCR). which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to